Welcome to 11th standard CBSE notes. The chapter excretion or the last part of kidney and excretion. Counter current mechanism. Kidney of higher vertebrates such as mammals, birds and man has ability of absorbing more and more water from tubular filtrate to conserve water and make the urine more concentrated. While the urine or filtrate from glomerulus, it goes to the tubules. Again, more and more water is reabsorbed. Why? Because the body want to conserve water or keep the water, good water in body. This can be achieved by a special mechanism known as counter current mechanism. The basic concept of mechanism of counter current are as follows. The Henle's loop and vasa recta, that is the capillary loop, play an important role in the mechanism. The flow of filtrate in the limbs of Henle's loop is in opposite direction and thus forms a counter current. The flow of filtrate, that is first is down, then is up. The flow of blood within the two limbs of vasa recta also occur in the counter current pattern. The osmolarity, that is the number of osmoles of solute per liter of renal cortical interstitium is the same, 300 milliosmol per liter, as in other tissues. Whereas interstitium of renal medulla is hypertonic with a gradient of hyperosmolality from renal cortex to the tips of medullary papillae. Due to this difference in gradient of hyperosmolarity, the water is conserved. That is, the water is taken inside in the cells. The hyperosmolality of medullary interstitium near the tips of papillae is as high as 1200 to 1450 milliosmoles per liter. Now we will see the mechanism. The gradient of increasing hyperosmolarity of medullary interstitium is maintained by a counter current mechanism and the proximity between the Henle's loop and vasa recta. So this gradient difference between medullary interstitium, Henle's loop and vasa recta, this all forms the counter current mechanism to conserve water in the body. The gradient is mainly caused by the to and fro movements of sodium chloride, NaCl and urea. The transport of these substances is facilitated by the spatial arrangement of Henle's loop and the vasa recta. There are two aspects of this mechanism. First, the counter current multiplication by Henle's loop and counter current exchange by vasa recta. Two mechanisms. And what is the movement? NaCl. Of whose the movement it is? NaCl and urea. And two mechanism, multiplication and exchange by Henle's loop and vasa recta. NaCl is transported by ascending limb of Henle's loop. These enter in the descending limb of vasa recta. See here. The afferent arteriole gets in glomerulus. From here, from the fine capillaries, filtrate is sent into this proximal convoluted tubule where the pressure gradient is 300. From here, descending limb of Henle, the pressure gradient is 400. Then H2O is reabsorbed in medullary interstitial fluid. This is medullary interstitial fluid, what is in white, and this is nephron in grey. Again it goes in descending, then ascending limb, where due to pressure gradients there is reabsorption of water, sodium chloride, again water till the last collecting duct. NaCl is returned to the medullary interstitium by the ascending part of vasa recta. But contrarily, the water diffuses into the blood of ascending limb of vasa recta and is carried away into the general blood circulation. Permeability to urea is found only in the deeper parts of thin ascending limb of Henle's loop and collecting duct. NaCl is in ascending part and urea is in Henle's loop and collecting duct. Urea diffuses out of the collecting ducts and enter into the thin ascending limb. A certain amount of urea recycled in this way is trapped in medullary interstitium by collecting tubule. The collecting tubule also plays a minor role in the process. The counter current mechanism helps in maintenance of concentration gradient in the medullary interstitium. Due to this counter current mechanism, even the medullary interstitium concentration is maintained. Presence of such gradient helps in an easy passage of water from the collecting tubule, resulting in the formation of concentrated urine or filtrate that is nearly 4 times concentrated than the initial filtrate formed.